So welcome to our discussion today about AI and clinical trial imaging. My name's John Himoff. I'm the Chief Product Officer here at IAG. I've been working in technology for a really long time, like 25 plus years. Working in AI probably even longer than that. Some of our experiences that we've done with IAG that I just wanted to share, because we learned a lot through them. We did a project on AI for histopathology, where we were looking at cell tissue samples and getting into the cell counting within the tissue samples and predicting the scoring on the Nancy index. And this was really interesting that we started with POC and worked through several iterations of it, and then we're able to generate a reliable application that went through a lot of testing and validation to actually count all the different cells within the tissue samples. And then we moved on to another project that was related to looking at ultrasound images around the endometrium and figuring out with the clinical data, looking for biomarkers. So it was both a machine vision and a machine learning activity, which was super interesting. And I think we learned a lot about that. But Michael's going to touch on it, but getting the data, annotating the data, writing the validation reports, understanding how it works, understanding how sensitive is the data to different ethnicities and different sources and even different machines that acquire it. It's all becomes a very interesting and complicated topic. And thank goodness that we have AI to help us through this. I want to introduce Michael because it's always a pleasure. Michael's really so generous with his time to share his ideas. I heard you were a big sensation at the conference recently. So thank you for reprising some of your slides for this webinar today.
Thank you very much, John. My name is Mikael Boesen. I'm Professor of Radiology at Bispebjerg Frederiksberg Hospital, Copenhagen University in Denmark, but I have known Olga, the founder of IAG, since her PhD time, and I followed them ever since. I have a small role in the company today, also as senior therapeutic area lead and advisor to the scientific board as well. So I've been working with musculoskeletal imaging since the very early millennium change, and also following the AI wave through several iterations for image generation over now visualization, segmentation, and prognostication, and the newest kit on the block, large language models that I work with both scientifically and also clinically. In my professional role in Copenhagen and also through the IAG portfolio. So I will share some of these experiences, and that's why I think the basics that we'll also come to is that why is it, and let's say the part of having this meeting, why is the clinical trial infrastructure such a good environment actually to use AI today? Because you need a strong foundation and audit trail, QC string, and data cleaning and high data quality in order for AI to work properly. Because if the data changes or you don't have control of that, then the AI will drift over time without you even noticing it. And this is what the infrastructure is built for. So you need that foundation to stand on in order to make proper clinical trials today, but also make AI work in the real evidence in the clinical setting, which is a completely different beast in that sense, which I will touch a little bit upon while we are going through the slide deck. It would take around 40 minutes to go through it, and we will leave some room in the end for questions, but please provide your questions in the chat box, and we will take them there, or which is easier because open the microphones and all that. I think it will not be feasible also for the quality and the agility of the meeting. Having said that, John, I think we need to start, and why don't you just take over here?
So just a few quick words about IAG. So IAG is an imaging CRO organization, and we've been working for the last 20 years with biotechs and pharmas to help them drive imaging into their clinical trial results. We took I think a leading step in the industry and built our own platform, which we call DYNAMIKA. And this is a specifically built for imaging CRO. It's the end-to-end solution for everything where the data comes into the system, is QC'd, is managed through the reading paradigms. We manage the data export, and of course, all of this is done on an enterprise scale with security and industry-level compliance for how we put the whole system together. And it's a big undertaking, and it's a high-performance, scalable system that can handle the needs of small studies, early small phase 1s that maybe have some unusual requirements, all the way through high-volume phase 3 studies. We've done studies worldwide, recently started working in China, so we have some good examples of bringing data out of China and well as collecting data in China. Studies really all over the world as the biotechs and pharmas continue to look for new areas to find patients. We're there picking up the data and bringing them in for the central reads. And I think ultimately that means that we're able to handle really any kind of study on any scale with very fast startups, and our system doesn't require any particular hardware or any kind of unusual setup. And you just need to have a browser to access the system, both for the readers as well as for the sites. So yeah, I won't go too deep into it. Very proud of the system. Just to give you a quick schematic of it, we can take in any kind of data from anywhere in the world. Typically it's DICOM, but it could be all kinds of other stuff. It could be simple spreadsheets, could be ultrasound, cine loops, videos, photos. Anything, any artifacts that are needed for the study, we can bring into the system. We then bring it in, and the system will automatically work through pseudo-anonymization, so the data won't have any identifiable information for the patients. Our team looks at the quality control that is very specific to the study. They then make sure the data is going to be usable. If there's any issue, they generate queries back to the sites and track those through. We then complete the QC and set it up for the reads. And those reads can be done by expert readers, Michael, as well as other experts anywhere in the world on their computers and multiple screens and in convenient as possible manner. We have a data management team that manages the data exports so that it matches the required formats for the study, working with study teams and creating data in all kinds of different formats and file types. One of the things we've done is to make the system scalable is we have a configuration for each study. So each study can be set up very specific to how it needs to operate. We have a task manager that drives the workflow, and really the study can be both flexible and scalable. That's really our mission with designing the system. Of course, as you would expect, all kinds of output reports, all kinds of reports distributed to all the various stakeholders, sites, PIs, CRAs, sponsors. We can send specifically the information they want to them. The system, we manage it ourselves. I think it's unusual in the industry where you have a technology team, and we're really technology people who manage the full design, build, run, support loop for the system. Just in terms of the AI, because that's our topic for today, one thing we haven't put into DYNAMIKA is AI specifically. We've designed the system to be AI ready. So we can plug in AI and use APIs or other data exchange techniques to leverage models that are coming out, whether we build them or Michael's team builds them or they're come from anywhere in the world. We can plug these AI tools into the workflows. And that's been our approach because that allows us to, to manage all the compliance and audit trails and have a really solid, reliable framework, but also have the ability to bring in new tools as they come on market and have proven themselves. So just how that fits in, the technology layer is what enables our end-to-end services. And as an imaging CRO, we go the full length to help sponsors not necessarily rewrite their study protocols, which is the last thing anyone wants to do, but to go into these protocols and find out really how should the imaging be delivered and implement and optimize the best way to do that. working with regulatory strategies and understanding how the FDA wants information. We have experts to help design and set up the studies at the delivery level. Yeah, I mean, I think a lot of information here on this slide. I don't want to go into all of it. I think the main thing is that IAG is really the end-to-end solution for the whole imaging requirements for the types of studies that you'll see Michael touch on. We work in the industry with all kinds of sponsors and biotechs and pharmas. Okay, so Michael, over to you, please. And again, we're very lucky to have Michael sharing his insights, a leader in the technology and in AI for a long time. So please go ahead.
Thank you, John. So that leads us to the agenda of why infrastructure, and specific, in my opinion, clinical trial infrastructure is the foundation for AI in clinical trials. So go through what AI is today. So what is a little bit about the landscape today? What is some of the current use cases where we already have some added value or potential value? So how it can reduce variability in read and in speed of reading, and then also inclusion in trials. And that leads to kind of why we see this as a target that it's coming regardless of whether we want it or not. So how can we move forward to quantify imaging endpoints, trying to avoid all the pitfalls, ensuring that whatever is used is very thoroughly tested and serves the validity of what you expect, control and handle the biases, the use cases and the governance, as well as some pitfalls that I will show you that are especially let's say affects the performance, and then ending with some strong implementation looks like in the trial environment, examples for where we can use it already today, and then finally John will, let's say, close it with some sponsor checklist there, what you need to think about as a sponsor in these trials. So having said that, there are three forces shaping rheumatology imaging today in clinical trials, and I will take you through some of them, because you first of all need to know that imaging is not a standstill market. It's a very fast evolving, very high tech, and has always been a digital tool that doesn't stand still long. So we have, for the last 20 years, gone from studying structure of anatomy with imaging methods like X-ray, MRI, ultrasound, and CT scans, to ultrastructure now coming into microscopic levels of visualization. With the newest generation of CT scans, for instance, we can go beyond 100 microns, sometimes down to 50 microns of resolution in the image. But that also comes with a lot of data, and we see something that we didn't see before because we couldn't see it. And then we also know, and I have to acknowledge that especially in the domain that IAG works in, all the indications, cancer, muscular imaging, immunology, perfusion, inflammation is the key driver, and understanding inflammation in the specific diseases and the change in inflammation upon an intervention or drug or even a placebo is important to understand the disease behavior. Leading also into metabolism, that there are overlapping arches here where you, from the clinical nuclear medicine approach, actually can begin to see how certain molecules at specific targets is affected and acting. So all of this is continuously evolving, and as data changes, so will the performance of the AI that you are using. So a multi-imaging trial could look like this. So in osteoarthritis, we now see today that we combine the conventional X-ray with a lot of other technologies, ultrasound, CT for inclusion, maybe for mapping changes over time, conventional MRI giving contrast to map inflammation, and then while giving contrast doing dynamic contrast enhanced MRI, and then adding also PET and PET tracers to understand, for instance, bone marrow lesions. And the more complex it becomes, obviously also it's it's important to have specific sites and the site selection to be able to perform this. It also can see earlier changes, but it also complicates things. And this is what the AIG is a leading expert to help you with. So it runs smoothly, and if sites are not included that are not able to deliver that performance upfront. We also know that we are drowning in data. Right now, we are doubling healthcare data in Europe every 7 to 10 days. And we have done that, let's say, doubling the healthcare data ever since we become digital in the millennium change. But it's like, let's say, the analogy of one drop of water, then double every time. And then when you see that the glass is only a little full, next time, the next second, you see that it flows over. We're also seeing that, and it's especially visible in the clinical side where we are lacking human expertise. And as data increases, the gap increases. We call it jaws, because the human cannot keep up. And while keeping up, they are also trying to perform faster, meaning error rates and interpretations of images also increases. And that is something we need to mitigate in order to not become a bottleneck in the clinical side, but even more important in the imaging side. And as data also increasingly are floated to the human, then we are also seeing burnout and let's say backlogs of piles of data that not have time to analyze. And the people that needs to analyze it are the experts, and they're also the one who need to educate the new people. So that's kind of a competition you can't beat. So how do we mitigate this? This is where AI comes in. And what do we mean by AI? And for instance, in rheumatological imaging, that AI is this an overarching principle of machine learning as well as deep learning methods that can help you to either do fast acquisitions, reconstructions. It's very mature. We have in all modern scanners. Segmentations, it's also quite mature. There are many models out there. Few of them are regulatory approved, let's say having a CE or an FDA stamp yet, but they are very well used and developed in the trial and research setting. Detection and classifications, that's also something that can be done more and more optimized. It's established, not yet fully matured. It's external validation against or construct validity against, let's say, RAMRIS scores or PsAMRIS scores, and so is still lacking to a large extent, but the technology for doing it on the images is there. Quantification and measuring change, it is emerging, but still not fully optimized, meaning that you need always a human in the loop, which is also important to say here that AI never stands alone. AI, whether we use it for, will always have, in the trial setting as well as in the current clinical setting, a human in the loop that signs off whatever has been analyzed. So nothing is done automatic by AI, nothing is fully optimized in the sense that we are not in control of what is happening with this human oversight. And then finally, prediction, decision support. This is research, because we are now beginning to see big data, and big data also gives a better chance to do the subgroup analysis, seeing phenotypes that respond versus those who don't respond, and even establishing some endotypes, meaning that we understand why you are developing that specific phenotype. But that's big data, and are from research transferable soon to a clinical trial environment as well. So cutting it down, AI can improve the entire workflow in radiology and in pathology and in wherever you have digital imaging. But you need to be aware of the pitfalls, the biases, and all of that, which again leads back to my statement and the conclusion, as well as the headline of this talk, is that AI is perfectly fit for a clinical trial environment, simply because it's so controlled of what data comes in, validated, scored to highest of standards with 2+1 adjudication in many of the trials. So this is the perfect place to either start or introduce an AI in your system. We also need to ensure that what we're talking about in the digital pathway, because looking at a patient journey in a trial as well as in normal clinical workflow. So we can see inclusions criteria or referrals, then we need to make a vetting of what is needed for that referral that's stated in a protocol when it's a trial. And then we need to ensure that the protocol is consistent and also performed in all the scanners, that the scanners have been tuned to perform it. Then we do the scans, and this is where the scans, many of the scans come with AI reconstructions even today. That is not fully controlled, but the trial environment we need are controlling that. And then finally ending up with the image analysis where it's visible. So here it's more controllable by the human what is happening, because you can see it with your own eyes, and to the report or the dictation or the let's say the pre-filled reports that now have been more and more optimized. Radiology is leader in this field for all the FDA and CE mark approved devices because it's the same data formats. That's an absolute lowest denominator of success. If you don't have the same data format, forget about scaling in AI. So, and consistency, when you have the same data format, you also need to ensure that that data format is consistent throughout wherever you take up. So the scanners has to be tuned to this, that the acquisition has to be at the same thing, the parameters has to be the same, otherwise then AI will fail without you knowing it. Again, leading back to the fact that this is all done and overseen GDPR, ISO, and HIPAA compliant ways in a trial setting, not in the real life setting, as it is today. So what I will talk about is to say the acquisition of the scanning and the image analysis that are, let's say, more, that is the mature part. The rest we will save for when it's more mature to the trial settings. And then we have to look at, for instance, all the diseases out there. So we're looking at inflammatory joint diseases like psoriatic arthritis, rheumatoid arthritis. We're looking at axial inflammatory diseases like axSpA. Also, PsA here has some and emerging evidence. Rheumatoid arthritis even have some actual, let's say, appearances. We're looking into muscles, for instance, myositis or scleroderma, autoimmune diseases, lung diseases that comes with a lot of these, let's say, autoimmune diseases. So these are organs that we usually have not looked at because they're not within the domain that controls it. For instance, rheumatology seldomly are dealing with lung diseases, but there are big overlaps similar to abdominal diseases, are not used to have joint diseases in their, let's say, mindset. So this is where AI really can help to cross these barriers in a seamless way and in a controlled way. And the trial readiness I will talk a little bit about here. So when we look at, for instance, KL grading, which is an inclusion criteria in all rheumatological trials, it's feasible with AI today. There's several companies and also GitHub available algorithms that can do it. The literature is out there. This is an AI from a Danish company called Radiology, where they actually can both do the KL gradings, the OASA grading, the joint space narrowing, as well as the patella osteophyte scoring, which is seldomly used for inclusion. It's only the PA acquisition of standing semiflexed knees in the anterior or posterior view. So it's as good as the expert, accuracy. It works across centers in real life settings as well as in trial data, and it can uniform it. It raises the level of different expertises from non-expert to other orthopedic experts to the same degree to 3 professors of musculoskeletal radiologies. So it really is as a uniform, and the AI is robust, meaning that it has 100% reproducibility. Human doesn't have that. Expert has it around 90-95% of reproducibility, but still there are room for differences because these are scoring and grading that are no, let's say, ground truth about it. So this is why you sometimes say a 2, sometimes a 3, sometimes a 1, sometimes a 2. That is the human in the loop here. The AI does it the same way always. You can disagree with the AI basically, but at least it's fully reproducible. And again, it can be applied. We also see now you can do virtual CT scans from MRI, for instance, and this is a way of getting better access to grading something that MRI has difficulties to do. For instance, erosions in the sacroiliac joint. Then you can do virtual CT scans. That is a sequence. There's several methodologies. There are 3 different distinct methodologies. I don't have time to go into the differences, but this one is AI driven and also CE and FDA approved as a tool. You then also can do grading of spine segmentation as well as report generation. This works in the clinics, but could also be a tool for more uniform grading of spine diseases. Not many of them are done. It's more inflammatory driven spine disease. This is degenerative. But again, you also see that we also can see the paraspinal muscle, which seem to be important in many of different indications, calling the L3 SMI index, so the muscle sarcopenia index at L3 level. So that would be if I take here the laser pointer here, you will be able to see here that L5, this is the L3 level. So the actual view of this would generate something that look like this. So the muscle bulk here, also around the abdominal part, is predictive of a lot of diseases in oncology, in inflammation, in many other indications, long way from where the disease may manifest themselves in other joints. So this is really something that is worth looking at. Ultrasound images is also used as a tool. Now you can quantify the extent of synovitis with AI. You can map the cartilage and look at that. You have robots that can scan automatically, so even make more uniform acquisitions of these trials. So all of this is established in the clinical setting and ready to be taken in, if needed, into a clinical trial setting infrastructure. And these are some of the tools that are out there. CE mark tools going from lung detection of lung diseases on X-ray, also findings on the T scans on the lung towards the knees, segmentation of synovitis, quantifying inflammations. One of the preparatory tools from IAG with the DEMRIC scores based on dynamic continuous MRI quantification. You can same data. You can use the K-trans data. There's also there. You can do bone age, fracture detection, so forth. So all of this are established methodologies. They are ready to be installed, but they need to be externally validated on the data that is generated, because otherwise you tend to fall into the trap of thinking that it works and your data and realizing that it doesn't. So on a trial level values, it's important to have consistency, high throughput, and also quantification, more and more going towards segmentation and continuous markers, going away from categorical scores. That still is the reference standard and also the FDA approved standards. But as data emerging are coming in from many other indications, that is also seem to be changing the perception of what is possible. So AI can help you to reduce consistency or increase in consistency, meaning improve variability between readers. It can raise the level of experts or non-expert to experts. It reduces the variability and helps standardize the analysis over time. And throughput is important because many of the scanners have expensive time slots. So if you can do things faster and below 30 minutes, maybe even below 15 minutes, then that also will affect the budgets because you can have more patients through in the same time slope than we could earlier. And that also helps you to segment and pre-process some images that then can be visually or manually or human oversight approved in very time-consuming, tedious segmentation task, meaning faster turnaround time, and also that will affect also budget and time to results. For quantification, we need higher reproducibility, and we, as I said, going from categorical scores towards volume, and quantification of disease patterns that we couldn't do before because it was too complicated or was too extensive. And then, as you see here, we already have it today. You can do a conventional knee MRI scan in 5 minutes. That is clinical usable, but also can serve the basis of the needed standard sequences in a clinical trial of osteoarthritis. So you have time to add more advanced methods like T2 maps for cartilage, like DCR MRI for quantification of inflammation and perfusion, and all of that. So it leaves room. So you go below the 30 minutes magic, let's say, time for having everything at one shot. So this is already established and will be in many scanners already. So this is what you just need to know. And then we need also, again, to repeat myself, go from traditional semiquantitative scores. You all know them: RAMRIS, PsAMRIS, SPARCC, Kelvin-Lawrence, MOAKS, New York grading radiographs for actual SPA, to different staging in, for instance, lung diseases, towards AI-enabled quantitative analysis to pre-process the data, to segment the data that then can be used and approved by the human in the trial setting, if needed and when needed. You just need that your infrastructure is ready for it, and many clinical infrastructures are completely not ready throughout the world, whereas the trial infrastructure that DYNAMIKA provide is completely compliant and ready for this. You also need to know that when something is approved, CE or FDA approved, it doesn't mean that it actually performs well on your data in your real life setting, because many of these algorithms, when they're installed in the wild, meaning in clinical trials, outside clinical trials, people get disappointed about the performance. And the reason for that is because it now has to work in a different environment with different scanners, different software, different patient population, different prevalence of disease, meaning how often do you see the same disease. So if you change that prevalence and the AI for regulatory purposes is fixed and doesn't say anything about clinical validation or value, then you actually are reducing your sensitivity and specificity, that then will generate more false positives, more false negatives, and also so it will be kind of affecting the whole thing, giving friction instead of, let's say, assistance. And this again leads back to in the trial setting, in a controlled environment, this will not happen, because you have the needed infrastructure to monitor for this, to mitigate that in the infrastructure. And here's a paper that down here from Insight into Imaging that said that when we look at, let's say, the 6 intended use cases out of the 139 commercial algorithms out there, then only 7 comply to 3 of the 6 use cases. And again, it's stated that you need to have age, sex, race, and ethnicity in your data for AI, so we don't discriminate the AI performance. We have that when we're doing clinical trials. We don't have it in real life settings, and that will tremendously affect the performance in certain ethnicities. So be aware of that, and this is something that we can control and mitigate by having it in the trial data. So we also need to have a governance around it. So the concepts of how we conform the system to the data collection need to be consistent and overall same parameters, same. This has to be controlled for. Then we need, so that the training of the AI, whether you do it on your own data or you take in an AI that also has shown to work, we need to have an infrastructure where you check that it's valid, and you need to have clean reference data for that. That is generated in the usual reading setting. So this is important to have that. And then you need execution and deployment in a secure environment that also has an audit trail. and a surveillance to check for when an AI is beginning to lose performance because new data comes in that it hasn't seen before and need to be retrained. So all of this is within the setting of a clinical trial structure. But as John said, this trials infrastructure is not AI in the motor of it, but it has the opportunity to handle AI whenever needed. And then you also need to look at what is research, because a lot of research is there. A lot of what you see on conferences is research, and it looks good. It looks fantastic. It performs well on that specific data. But then there's a long way from having made that algorithm and written the code to we actually can include it into a trial setting as well in the clinical real life. That needs a lot of extra control, extra validation, extra secure, so we can ensure that whenever we use an AI, that it performs well, that it performs efficiently, that it performs to the scope and not deviating from the intended use case and performance. So these are just the summarization of that, where we need to be able to that and remember that just setting an AI free in the wild for optimization without the human in the loop, it's possible for some indications already, but we have tested it, and an AI also make errors because it's based on human input from the clinical side, and this means that we need to ensure that there's still human oversight regardless how optimized it has to be. So this optimization process kind of is not happening in the clinical trial setting. In the clinical real life setting, but in the clinical trial setting, it will never be fully optimized. It will always be pre-processing and human in the loop oversight. And then we can also play with the data, big data, subgroups, finding signal in the data that is already acquired. This is what we need AI also for, because this is where it really does its magic to help us understand behavior, patient selection, because patient inclusions are often very broad, very crude, and then we see treatment responses in some but not all, and we need to understand why this is happening, so we better can plan the next phases, and here AI can really help us with this data mining. And obviously, with all the data that we generate, we're drowning in it, so it's really also difficult, also in a trial setting, to get access to and use all the data that we generate. Here, LLM-based report summarization or findings patterns in the acquired data is also very, very useful and can be handled in an audited environment. So finally, as we also are recording and scribing this meeting with an ambient scribe, obviously this is also something that can help to take all the notes that needed and summarize it when they're in negotiations, and also talks about follow-up points in many of the clinical trials. So soon to finish up, we need to say that AI trial grade workflow needs to be the same control as any other regulated trial data set. So whenever you think about and implement AI, this is important, that we need to have a version log model, and that model we need to overview. We have the full audit trial. The pre-trial validation is important before we run it on data. And then obviously it has to comply with all the regulatory requirements, but this is given since it's in an audited trial environment. We need to find out how it should use. Should it be a single reader, or should it be, let's say, a second reader or first reader, and with 2 human adjudicators? That is for trial to trial to decide. And obviously we monitor the drifts whenever an AI is installed, because when data comes in, we need to ensure that it can still be read by the AI, and also whether the AI is not compliant to what is the human ground truth, and always having the human reader in the loop again. And then we have some examples, MRI synovitis self-to-review for AI. So there are several indications where it has been used to help granulate data in rheumatoid arthritis, automated joint segmentation, synovitis volume estimation, endpoint mix, and the governance is always there. So it really have shown that AI can also help us to see better separation and dose response than human readers alone, because you go from categorical scores to continuous scores. For instance, this is a paper that we did with one of our collaborators. So it is possible, and a lot more that we will take up when it's more ready. But now, because the time is also at the essence and we need time for questions, think we need to wrap up now. And John, please, if you take it from here, and then we are happy to take questions.
I mean, actually, Michael, I wanted to ask my own question, since I've been listening to what you're saying very intensely. I mean, I think you did a great job covering kind of the landscape, and it seems very clear that AI is going to generate more data. It's going to process that data faster, and we have the potential for new insights. I hadn't heard before about the virtual CT, so that's really cool. That, like the NPRs that we're seeing with the reconstructions, that you can do new stuff and create different kind of acquisitions virtually. That's a super cool concept.
Yeah.
I guess the question I had for you, what does that mean for some of our sponsors? Do you think, and maybe this is the kind of multi-million-dollar question here, could they go back through their existing studies with these new tools and find new insights, or even find out that the drugs they were trying to bring to market performed better than they thought?
Yeah.
How do you see that happening?
Yeah, certainly. So first, to answer your first question about virtual CT is a specific sequence you need to do. So it's you can't use retrospective data
like the NPRs, right?
Yeah. You can't use the retrospective data on your current MRIs to do this yet. But so just to say that, so you need to do it prospectively in trials. But CT is coming in and looks extremely promising, also because it now has a very high resolution. You see things it didn't do before. But also, let's say, in osteoarthritis, for instance, you now begin to look at bone shape as a biomarker for progression of OA towards end-stage knee osteoarthritis and joint replacement, which is FDA's, let's say, end game of approving a drug that you can prevent joint replacement. So this is where CT is coming in. So we have done MRI forever, but CT is really exciting in many domains. It's not only in osteoarthritis, but also in inflammatory say arthritis, because it's the reference for bone imaging. There's also the whole concept now of sarcopenia, obesity, as well as trabecular bone, and what are the new GLP-1 drugs, for instance, doing to sarcopenia and osteopenia. It's well known that you lose mean muscle mass as well as bone while you take these drugs, which you also would do to some extent if you were on a diet, because you don't eat, so you do not get the nutrition needed and all of that, and you don't exercise. But the fact is, this is also add-on, and this is why it's so important always when you have an idea or mechanism of action of the drug, that we discuss what is the mechanism of action of the drug, and what are you expecting to get out of an imaging protocol. Because then it's better to advise, and with oversight of all these new emerging technologies, then we can advise to use what is required to a minimum standard, and then add something new that can separate the results from others that is in the field, so you get a market advantage. So it really is that. But also, having said that, the rheumatoid arthritis trial we did, that was a retrospective trial that failed with say categorical scores reading, but then when we optimized the synovitis grading to a continuous marker instead with the existing data, and actually showed there was a dose response suddenly in the data that we didn't see before. So yeah, a lot of the things that are stopped in phase 2 maybe that seems to have failed because the method that you used might not have captured the essence of what the drug did. Then AI can help you also within patients now, because we see from for instance now in oncology they're doing the basket trials or other more advanced or novel trial designs where you actually, if you fail one drug, then you go over into another trial settings having a new mode of action, because not one drug it works on everybody. So, but we also need to have something to offer to the patients that fails, right. Because then they just drop off the trial. And then what happens then? So this is a way to actually within the trial setting subgroup the responders from the non-responders, and then seeing new indications coming up or new molecules that might work better in the patient that fails. So it's a really good way of thinking about how we can maintain that audit trail and keep the patients in the loop for the next line of drugs that is coming, so you don't mix the failures with the responders. Right. If you can follow me.
No, I think that's right.
Because that's what we learned from oncology. And it's coming now also to immunology trials, into rheumatoid arthritis, psoriatic arthritis, this same concept of novel targets. You also see CAR-T that also came from lymphomas and hematological oncology into now rheumatoid arthritis. So it's emerging fields that we need to learn from what is done in other fields and translate that into novel ways of doing imaging and designing trials in other indications. But this is also what is good for because as we are a broad portfolio of the various indications, we have that opportunity to have that transfer of knowledge between trials, because we have the existing, let's say, expertise from one indication that we then can suggest and add to another indication.
Yeah, I mean,
did answer the question or raise more?
There was a big topic. I was in Boston for the ScopeX conference, and a lot of the sponsors were saying, well, wait a second, we have this huge amount of data from all the different studies, and actually there are cohorts that responded better. And I was like, yeah, but you didn't even look at the imaging data. So actually that's just on the kind of clinical data. If you could reread some of the imaging data, maybe you'd find that even more cohorts had better response than you thought.
Yeah, and even, for instance, in musculoskeletal, there's a rheumatoid and arthritis trial with the placebo group, because it's remitting, relapsing, especially arthritis. The placebo group is also always improving, because time is let's say against you when you do trials, because over time, when you go to a doctor have a certain pain, then over trial that pain will wear off and then come again. And the incident of that coming again may be higher in the placebo group, but you might hit them, let's say, when they are downslope, when you do your follow-up at 1 year or half a year. So that's why placebo always seems to improve, meaning it's very hard for a drug to be better when it's pain that is the primary. But here it's important to understand so what is it, what kind of profiles it is from the non-responders in the treatment arm and the or the non-responders in the placebo arm. Are they similar or the difference? And that we can use machine learning AI algorithms to actually help us to understand, to get that insight with existing data without generating new data that actually can fuel the understanding of why that drug might not work. And that will be added now to the inclusion criteria of the next phase. So we're kind of trying to select
for the design layer.
Exactly. So we're trying to select the best patients to the best molecule, personalized medicine, some would call it, but I think it's just reusing already existing data to improve your design onwards, and also the label that then will come, might be a lower group, but still it will come to market and have a very high hit rate of let's say making a difference, and responders, meaning that you will stay on the market as well. So
interesting times. You had one other comment which I really enjoyed that I just wanted to ask you about again. You said that overfitting is your friend. which,
Yeah.
when we're doing AI stuff, overfitting is never your friend, but
Nope.
in this environment, I think that was a really interesting comment. If you could just share that again.
Yeah, I will share that again, because you need to know that if the AI knows the data that has to work on it performs super. It's now almost have superhuman performance. It actually beats the human in the loop.
Yeah.
Because it's based on the data and it knows the data, and then it overperforms. If you took that AI algorithm and put it into a completely different setting, in a clinical setting or a different patient population in a different trial, that model would perform different and often poorer. But we use that fact as our friend in a clinical trial setting. So we ensure that the AI actually as sensitive, as good, and maybe even superhuman within a trial setting, because it has been trained on and know the data that has the same format, has the same model, has the same scanner, software, patient selection, all of that. So that's actually a very good thing in a trial setting. It's a very bad thing if you want to scale into multiple different clinical settings. And that's, I think that is a concept that sometimes is confused, because you mix what you know from the clinical side into what is known and what is used in the trial setting. That is not as open or visible to people that don't work in it.
And I think also some of the multi-agent approaches, kind of you want to have overfitting, because you could have different flavors of an AI that were targeted for different cohorts.
Exactly. And that again, and for the people that don't know what overfitting is, again, to narrow down the concept, maybe you are better at explaining that, John. But as I see it, it is that you know the data already, meaning that your performance, just by that fact, is inflated.
Yeah. Right. Yeah.
But that's a good thing in this setting.
That's a good
because
thing.
You need to be able to be granular, responsive, because it adds granularity, it adds responsiveness, which we like in a trial setting. So that's the difference between real life and a trial. Again, coming back to the conclusion of this meeting, I think would be that also what was the headline of this talk, that a clinical trial infrastructure with the audit trail, with the compliance, with the let's say clean data flow, with the highest reference standards, is the perfect fit for running an AI. And I hope that what we went through in this hour now has, let's say, ensured you that this is the case.
Yeah, and I think it's absolutely right, and that actually doing this and creating AIs for trials is much, I don't want to say easier, because it is very complicated and very intricate work, but it is faster than it's ever been. And if you have all the pieces to assemble it,
and you don't need that many data, because your sample is narrowed down to a specific target, a specific indication, a specific, let's say, population. So that's why it's you don't need thousands of data or millions of data points that need to cover everything in the wild. And by that I mean in the clinical setting. For a trial endpoint, you just need a few hundreds or even less.
Great. I don't know if any other questions. I can't see the questions list. Anything else come through? I don't even know how I would see that.
Can anyone see the chat? Let me see here. Chat, there's some.
Okay, I see the chat. Okay, I got one question here. Can AI correct for images that are not collected well as per specifications, i.e. mispositioning of hands for X-rays?
Nope, not yet. Hands for X-rays, no, not yet. For reading KL grades, joint space narrowing, yes. But not for X-rays. And this is why you need the expertise and the QC.
Right.
And the training.
Positioning devices.
And the position devices
train
and all of that.
The
sites need to be aware of that. A few people, we need to be validated, need to have to go through, let's say, several steps to be approved for that. Need to send, let's say, a test case, and we will continuously monitor that while acquiring the data, and also return back because it scores after quality when the quality is not matched. And if it happens continuously, then it flags just 2 or 3 times, then it flags a retraining acquisition that then is followed up on.
Yeah, automated response.
Yeah. Exactly.
Okay, well, that was a great hour, Michael. Thank you so much for your time.
It's always a pleasure. Yeah.
Super interesting. We're going to share this recording, excerpts of it on our website in case anyone wants to come back into it. And of course, if anyone out there is interested in working with us on AI-enabled workflows, you have all of our contact information, and we'd be very happy to discuss future studies or even looking at existing data that you might have and find out new insights from data you've already collected and retrospectively want to learn more about. So that's great. So thank you again, Michael. Thank you guys all for attending. And we look forward to our next webinar, which should be at some point in the next month.
Thank you so much.
Have a good afternoon, everyone. Good evening.
Yeah, bye-bye.